Hello, everyone. So I would like to give you a brief overview of the poster I'm presenting at ISDN that is entitled Oligodendrocyte Precursor Cells Guide the Migration of Cortical Interneurons in the Developing Cerebral Cortex. So as you may know, OPCs are generated in three distinct waves in the forebrain. The first one generated very early at around P2.5 from preoptic area and the medial ganglionic eminence, followed by a second wave that is generated by the lateral ganglionic eminence and also caudal ganglionic eminence. And finally, the cortex itself will generate the final wave of OPCs that will populate the entirety of the cortex. While the function of the third wave OPCs is more clear, we don't have much information about the functional significance of first and second wave OPCs in the brain. So in fact, in this work, we showed that these ventrally generated OPCs from the first wave lead to a disruption in cortical interneuron migration, as you can see here in these images. So in fact, when we deplete the ventral OPCs, you have a disorganization of distribution of interneurons within the ganglionic eminences, and also a reduction in the number of interneurons reaching into the cerebral cortex. We investigated in more detail the mechanism associated to this phenotype, and we realized by performing time-lapse video microscopy that in fact, in the absence of OPCs, interneurons will start interacting with high affinity to blood vessels that are on the way. And this interaction of interneurons with blood vessels is a factor that delays the arrival of interneurons into the cortex. So how does it work? In fact, these blood vessels are covered by these first wave OPCs. And in fact, when interneurons are passing close to blood vessels, there is a contact repulsion mediated by OPCs towards interneurons, and interneurons will move away from these blood vessels, and so they can keep progressing in the cortical parenchyma. And we did several experiments that allowed us to understand why would OPCs perform this contact repulsion with interneurons. And we figured out by playing with the levels of 6L12, which is a chemokine that is an attractant for both populations, that in fact interneurons are attracted to blood vessels because endothelial cells release 6L12, but 6L12 is also an important chemokine for OPCs to regulate their migration and proliferation. And in fact, when OPCs are present in blood vessels, interneurons are no longer able to come and interact with these structures to sense 6L12. And we found this by using antibodies blocking 6L12. Another strategy that we used is, in fact, detaching OPCs from wild-type slices. And in fact, the detachment of OPCs from these structures promotes the interaction of interneurons with blood vessels, suggesting that, in fact, the two populations are at competition for the same chemokine, 6L12. So in conclusion, our study shows for the first time that first wave OPCs have a very fundamental role during development in guiding cortical interneuron migration by preventing their interaction with blood vessels, a factor that would delay their arrival into the cortex. Thank you for your attention.